guys, welcome to Bio 112 Online, your home for biology education. Today's topic, the immune system. We've come to the part in our series where we need to talk about clonal selection. Clonal selection is going to be the rapid increase in the numbers of lymphocytes with receptors that are capable of binding an antigen. This all starts off early on in development. During development, the creation of a diverse array of lymphocyte receptors provides the body's ability to first recognize a diverse array of antigens. Second, it allows the body to have a lack of self-reactivity, so the body does not attack itself. And finally, it allows for the formation of immunological memory. So how does the body go through and do this? Recall as we were talking about the different types of lymphocyte receptors, the B cell receptors, the T cell receptors. In both of these cases, we had areas of the proteins that were constant, that were the same amongst every B cell and every T cell. In addition, we had what we referred to as the variable regions. These variable regions will be the regions that are capable of recognizing the antigen and are different between each T cell and B cell. So, how is it that you have some cells that are all B cells and all have the same genome, that have the same essential protein, but some portions of that protein are inherently different in the variable region? It all comes down to during development when you have mutation. That mutation is not the kind of random mutation we talked about last semester. It's a very specific type of mutation that's going on in a very specific region of the gene. While each B cell will have the original gene for the B cell receptor, in the process of early development, you start seeing lots of mutations occurring in that non-conserved region. This level of mutation is quite deliberate because it means that each subsequent B cell is different from those around it in terms of that region. When those genes are then transcribed and translated, it means that the proteins involved will have slightly different binding patterns for their hydrogen bonds, which means that each one has a slightly different assemblage of hydrogen bonding points, allowing it to recognize different antigens. So for example, for a B cell that might start off initially the same as all the others, you may have some mutation in that variable region. So now that the B cells start becoming slightly different. With that differences in that variable region, it means that their receptors are now able to recognize different antigens. As lymphocyte development progresses, you can end up with a population of cells with a very large diversity of receptors. And that diversity of receptors can recognize almost any disease that you can imagine. As a result, when you're born, you have B cells that are capable of producing the antibodies for smallpox and for rabies and for many other diseases that are considered to be deadly and dangerous to you. That sounds like very good. But unfortunately, you're also born with B cells and T cells that have receptors that will recognize and attack epitopes that are also naturally part of you. This creates a problem. And the solution to this second problem is going to be the concept of self-tolerance. Self-tolerance is maintained by eliminating those B cells and T cells, those lymphocytes that have receptors that recognize parts of you. This occurs early in life. While later in life, when you have a lymphocyte that is activated, it finds something that fits its receptor, it then goes on to divide and become active. In early life, lymphocytes with receptors with antigens that are already present in the body and become activated then undergo the process of apoptosis, which means that they undergo programmed cell death and therefore are eliminated from the body instead of becoming activated. This becomes the first step of clonal selection. Clonal selection, as you recall, is going to be that rapid increase in numbers of the lymphocytes that are capable of binding an antigen. So how does that really work? Well, this is kind of what we've talked about already. Anytime those lymphocytes become activated, they begin to divide. Now we have something interesting going on here because it begins to address one of the problems we pointed out early on in lecture number 1. Recall, for microbes, they evolve much faster than people do. We have a generation time of 20 years. They have a generation time of 20 minutes. So how do humans keep up? Well, some of the key cells responsible for maintaining the body's immune system have all the requirements that we normally think of for evolution. You have receptors that are responsible for binding those antigens. But initially, at birth, you have a vast diversity of those, and their cells are variable. In addition, you then have that variability being conferred by differences in DNA, which means amongst the cells, that trait is heritable. And then once those cells become activated, they have a tendency to divide more quickly than other similar cells, which gives them differential reproductive success. So therefore, while it may take 20 years for you to have a generation time, your body's immune system's ability to evolve happens much, much faster because it has all the main components for evolution to take place. This occurs in both the humoral system, those B cells that are going to be responsible for producing antibodies, and will also be present in the cell-mediated system, in which case you have cytotoxic T cells, which are responsible for finding out cells that have become infected by bacteria and viruses, or have begun to malfunction. So, for example, an activated killer T cell will rapidly replicate into many identical cells, or what we refer to as clones. Each of those clones will have the capacity to then go forward and actively fight that infection. However, we also have this concept of memory. It's not just simply about proliferating the cells that will enable the body to fight that infection. You also have an increased number of non-active lymphocytes that can bind to that antigen. As a result, while you have this proliferation of those killer T cells, some will go to fight the infection, but others will stay behind and serve as memory cells. The net result of this is that you have a much larger population of lymphocytes with the receptors necessary to fight the infection the second time, or the third time, or however many times you encounter the same antigen. The same is also true for B cells. If you have an antigen present in the body, you may have the B cells that can recognize that antigen and have the ability to produce that right antibody. Some of those B cells will then go on to replicate and become plasma cells. However, the rest will become memory cells. Those memory cells will stick around, and the net result is that you have many more cells in the body that can produce the right antibody if you need it at some point later. This is going to be critical. Activated B cells and T cells undergo clonal selection, producing effector cells that are short-lived and immediately attack the current pathogen infection. However, you also have the production of memory cells that are long-lived and that can later be activated by the same pathogen or a similar antigen that triggers their formation. We can refer to this first event as the primary immune response. The primary immune response occurs in response to the first exposure to any antigen. It usually peaks in about 10 to 17 days after the initial exposure. This is going to be due to the production of those effector cell clones. But it may take a while to get started. The secondary immune response is the response to the second exposure or subsequent exposure to that same antigen. It is faster, on the order of 2 to 7 days. It is bigger, and it is longer lasting. It depends on those long-lived clones of B and T memory cells from that initial exposure and from those subsequent exposures. That much longer response means that you innately have the ability to fight the infection. The interplay between that primary and secondary response is going to be critical for the ways in which we view and treat communicable diseases. For many diseases, as we said, such as typhoid fever, for many diseases, such as we said, like smallpox, like rabies, like Ebola, like SARS, in each of those cases, we have the ability to fight those off, but the amount of risk that our body has is going to be due to the amount of time it takes for that disease to be present versus how long it takes for our body to go and respond to it. If the disease can happen faster than that 7 to 10 day period that it takes for our immune system to get up and functional, then it means that the disease has a chance to win. However, if our body can get up and going faster, then our body has a chance to win. This inherently is also why we're very concerned about things like vaccines, which will go through and present an antigen to our body, which will then allow our body to go through and do that primary infection when there's not an infectious form of that pathogen. Therefore, the next time our body encounters that antigen in the form of the actual pathogen, then our risk of having a negative reaction to that pathogen is going to be much significantly less, leading to our body's ability to save our own lives. The proliferation of helper T cells, cytotoxic T cells, and B cells allows the acquired immune system to effectively deal with a current infection, and it also allows the body to recognize that same pathogen in the future and respond in an even more robust fashion. And that's it for now. But tune in next week when we will be covering the cardiac and circulatory system. So until then, stay safe, stay healthy, and I'll see you later.